Welcome back to pathology. Today we are going to go through the genetics of tumors. So the epigenetic changes are either methylation or acetylation, where there is going to be the silencing of some genes and the activation of other genes. The important ones we want to pay attention to are the RAS and the MYC genes. The decreased tumor suppressors are going to be BRCA1 and 2, so that's breast cancer, along with the P53, which is associated with Li-Fraumoni syndrome, and APC, which is associated with the familial adenomatous polyposis. The mutation of chromosomes 2 and 22 is associated with Bcell lymphoma, and the translocations of chromosomes 6 to 11 is usually caused by HPV. So the RAS gene makes the RAS protein, which is going to cause intracellular phosphorylation. Normally, it's going to be regulated by the growth factors, right? And from there, it activates a transcription factor, which allows the cell to go from G1 into the S phase. In a mutated RAS gene, it's going to make the RAS proteins that are already activated, so the cell cycle can just continuously go. The MYC gene on chromosome 8 normally makes the proteins for cell growth and survival, and then the activation of the MYC gene without regulation is going to cause uncontrolled cell growth. In lymphomas, the MYC gene is overexpressed or unregulated. P53 is going to be stimulated by damaged DNA. It's our tumor suppressor gene. It's a transcription factor, and it makes P21 to arrest the cell cycle by inhibiting CDK in the cyclins. It's a twofold kind of process. So it inhibits the CDK in the cyclin in order to stop the cell cycle, to allow the cell to repair itself or judge that there's too much damage and go through apoptosis. It upregulates the transcription of the inhibitory kinases and prevent the propagation of those damaged cells. So by stopping G1, it uses GADD45 for DNA repair, or BAX pathway for apoptosis. The mutation in P53 or suppression is caused by HPV. Li-Fraumoni is responsible for the germline mutation of the TP53 gene, and that is going to cause a predisposition for all cancers because there's no stopping the cell to judge whether it needs to go through apoptosis or not. More commonly, it's renal cell carcinoma, but it's not limited to just that. So the cancer cells are pretty nefarious because they make their own growth factors. So on one hand, they make all of the RAS proteins that aren't going to be activated by growth factors, or it makes their own growth factor, so it will stimulate the normal RAS proteins. And these RAS activated proteins are going to make the DNA polymerases bind to the promoter regions, and in cancers, there's either an excess amount of growth factors or there's going to be mutation of GTPase, which will continuously stimulate the RAS pathway. Neurofibromin protein is going to be mutated in neurofibromatosis type 1, and myelin is going to be mutated in neurofibromatosis type 2. So these are going to allow the fibrotic tissue to grow around the neurons without any regulation. So the retinoblastoma gene 1 is a tumor suppressor gene, and it makes the retinoblastoma protein, and that blocks the cyclin D and by binding and inactivating the transcription factor E2F. So in doing all of that, it stops the cell cycle at the G1S checkpoint. And the mutated Retinoblastoma 1 will allow the retinoblastoma to be phosphorylated and the cell cycle to just continue through the checkpoint. WNT is a receptor that signals the beta-catenin to bind to DNA. So beta-catenin is going to regulate and coordinate the cell-to-cell adhesion and the gene transcription. So, of course, if mutated, it's going to cause like the clumping of the cell. When cells clump, it can spread the the mutated proteins or mutated path like a lot faster. So how does that happen? APC is going to degrade the beta-catenin and it controls how often mitosis occurs. And without that regulation, the beta-catenin is allowed to proliferate without signals. It's going to be particularly mutated in FAP, hepatocellular carcinoma, colorectal cancers, breast, ovarian, and endometrial cancers. So FAP, familial adenomatous polyposis, is going to be an autosomal dominant inactivation of the APC gene on chromosome number 5. There's going to be thousands and thousands of polyps, as you can see here. And it's associated with Gardner syndrome, where the gastric and small intestine have cancers, along with osteomas, retinal lesions, and fibrous tumors. It's also associated with Turcotte syndrome, where there are medulloblastomas or glioblastoma multiforme. The peduncular or tubular polyps are rarely malignant. We have to be careful of the sessile or villus ones because these can proliferate. So if they're greater than 3 centimeters, they have to be removed by surgery. If not, it can cause vegetation to spread and accumulate on the heart. Okay, so Ataxia telangiectasia is an autosomal recessive disorder where the Ataxia telangiectasia gene is mutated. It causes abnormal cell death in the brain that helps with coordination and movement, so that's going to be more in the cerebellum. The ATM protein normally detects the damage in DNA, but if they allow it to continue with the cell death, there's going to be problems with movements, so there's jerky eye movements, also known as nystagmus, the ataxia, so unsteadiness, and seizures. So unfortunately, this presents young, and there is slow or decreased mental development that stops after age 10. It's characterized by these café au lait spots after the child has been exposed to the sun, along with premature gray hairs. And these children generally die because of an opportunistic respiratory tract infection. Bcl2 is Bcell lymphoma, and Bcl2 gene is going to code for proteins that regulate apoptosis. Isoforms 1 and 2 are going to bind to BAD and BAK. So BAD and BAK is going to be apoptosis. Proteins that allow cytochrome C and the reactive oxygen species to leak out of the mitochondria. Caused by the translocations between chromosome 14 and 18. And when we look at the histological sides, there's different sizing of the follicles. Okay, so mantle zone lymphoma is caused by translocation from chromosomes 11-14. Cyclin D1 is going to be overproduced, and that's going to independently cause the cell proliferation and differentiation without regulation. Centroblastic lymphoma is usually lymphoid polyposis, and that's usually seen in the colon around the ileocecal valve. So this is going to be the sessile villi. Ewing's sarcoma is going to be caused by chromosome translocation between 11 and 22, and there is going to be bone marrow problems. HPV is a DNA virus. There's 5 types, and they synthesize E6 and E7 oncoproteins that inactivate P53 and retinoblastoma. So the P53, once again, is going to regulate the apoptosis, and the retinoblastoma is going to allow the cell to go through all the checkpoints. There are the episomal forms of HPV, which is the more important form because that integrates into the host genome, and that can like spread the cancer. The HPV filiform projection, it's in the skin. It's called HPV striatum, where there's all these like cutaneous lesions. And then HPV latum, or the flat form, is going to be caused by syphilis infection. They will affect the stratified squamous epithelium, especially in the GIT, and especially in areas where patients have sex. So there's going to be the oral cavity along with the anus and the vagina. So how do we test it? We find the koilocytes, and in them either the keratin pearls or the intracellular bridges using a pap smear. EpsteinBarr virus will cause Hodgkin's lymphoma, and they are going to be characterized by the Reed-Sternberg cells via the CD21 receptor. So a nice little like memorization technique is you must be 21 to drink in a bar. So 21 is going to be your marker, CD21, and then it's Epstein-Barr virus. Okay, so the Hodgkin and Reed-Sternberg cells are going to be clonally infected because the Epstein-Barr virus is episomal. So it starts with one bad cell and it proliferates, unfortunately. And the proliferation of the cells, there's going to be a overexpression of LAMP1, which is going to be a TNF receptor, and that activates the NF-kB and JAK-STAT pathway, and that upregulates the anti-apoptosis gene. So another variant is HTLV1, and that causes T-cell leukemia lymphoma. Burkitt's lymphoma is caused by Epstein-Barr virus, and there are different types, and all of them have a relation to the c-Myc protein, and they have a starry sky appearance in the bone marrow, and that's where these macrophages are going to be filled with these tingible bodies, as you can see here, and they're surrounded by all these like small blue cells. That's why it's a starry sky. Hepatitis B, the gene can be direct or indirectly activating the transcription factors and signaling transducing pathway. So it's caused by long-term liver cirrhosis, and the cells that are affected are going to have these Mallory bodies. They can either have the trabecular, as you can see here, the acinar or solid or cirrhosis patterns. And one of the good markers is the alpha fetoprotein. Okay, so the colon cancer marker, CEA, alpha protein, is going to be a marker for liver cancer, and these are going to be your oncofetal protein antigens. These are non-specific, really, so we really use it to follow up after surgery to see if there are any cancer cells like left behind still making these oncofetal antigens. So the differentiation specific antigens are going to be your CD10 and your PSA. So CD10 for the liver cancer, PSA for the prostate. SIADH is going to be causing the brain edema and the hyponatremia. The carcinoid syndrome is going to make the products, especially histamine, serotonin, asthma, diarrhea, peptic ulcer, fibrogenesis in the right heart, thanks to the vegetation. And then there can be polycythemia. Lymphoma is going to be B cells found in the thymus. There's cancer of the thymus. It's causing myasthenia gravis. So one of the treatments of myasthenia gravis is actually just a thymectomy. But particularly in children, it's going to cause immunodeficiency. and they can undergo extensive hemorrhage, necrosis, and cystic changes, and form a nice little goiter. So angiogenesis occurs in all the tumors, but all of that eventually leads to the central necrosis due to the disorder of the angiogenesis. There's like all these railways, right? But nothing is straight. It all loops around, and there are places that are not going to have any substance, of course. They're increased levels of VEG and BFGF is going to, of course, lead to more angiogenesis. TNF alpha is an inflammatory cytokine, so that will lead to necrosis or apoptosis. And there are no cytokeratin stained cells in the peripheral blood of like all the cancers, because there shouldn't be any epithelial cells in the blood, because blood is the mesenchymal origin. So if you find any kind of like epithelial cell or cytokeratin stained cell, you have to immediately suspect some kind of cancer. The oncoproteins are high in the like liver cancer and whatnot, because the tumor cells will home in to the the tumor, even if they're produced by somewhere else. Like, let's say the bone marrow is like affected, it's going to make all these tumor cells, but let's say the tumor is going to accumulate in like the prostate, in prostate cancer, so they're going to be produced in the bone marrow, but then they're going to travel to the prostate. We're not really sure how this works, but we do know that they are affected by these oncoprotein levels. That's it for now. Thank you for your attention, and good luck studying.